complement system is an essential part of the innate immune response, playing a critical role in defending the body against pathogens. It consists of a series of proteins that work together to identify, tag, and eliminate invading microorganisms. Complement activation can occur through three pathways: the classical, lectin, and alternative pathways. Each pathway has a distinct trigger, but ultimately converges to the same end goal: the formation of the membrane attack complex, which destroys the pathogen. Let's explore these pathways in detail, starting with the lectin-dependent pathway. The lectin pathway of complement activation is triggered when mannose-binding lectin, or MBL, recognizes and binds to specific carbohydrate patterns containing mannose present on the surface of pathogens. This binding activates MBL-associated serine proteases, or MASPs, which then cleave complement proteins C4 and C2. C4 is cleaved into C4a, a small fragment that diffuses away, and C4b, which attaches to the pathogen surface. Similarly, C2 is cleaved into C2a and C2b, with C2a forming a complex with C4b. This complex, C4b2a, serves as the C3 convertase of the lectin pathway. The C3 convertase cleaves C3 into C3a and C3b, with C3b amplifying the response by depositing on the surface of the pathogen, ultimately leading to the formation of the C5 convertase and activation of the terminal pathway. The lectin pathway is a powerful mechanism for recognizing and responding to pathogens without the need for antibodies. By activating the complement cascade, it amplifies the immune response and contributes to pathogen elimination. Alternative pathway. The alternative pathway of complement activation functions independently of antibodies or lectins. It begins with the spontaneous hydrolysis of complement protein, C3 into C3b. C3b binds to hydroxyl or amino groups on the microbial surface, marking it for further activation. Factor B then binds to C3b and is cleaved by factor D into BB, forming the C3b-BB complex, which is the C3 convertase of the alternative pathway. This complex can cleave additional C3 molecules, generating more C3b fragments and amplifying the response. Properdin, a regulatory protein, stabilizes the C3b-BB complex, increasing its efficiency. As more C3b binds to the C3 convertase, it forms the C5 convertase, C3b-BB-C3b, which cleaves C5 into C5a and C5b. This marks the start of the terminal pathway, leading to the destruction of the pathogen. The alternative pathway provides a rapid and robust defense against pathogens, operating independently of antibodies or lectin recognition. By continuously amplifying the response, it ensures an effective immune defense.
Please like, share and subscribe, and don't forget to hit the bell icon for more videos.